Okay, this video lesson is to kind of go over some of the material that talked about or covered in terms of the development of the microscope and different types of microscopes. We'll use the timeline that was created in order to kind of touch back to the different people who helped contribute. There's more people, but we're focusing just on a few of them. So the first is Roger Bacon. And Roger Bacon, you can watch the video if you like, but he did a lot of work with lenses and mirrors in the 1200s. So around 1250, he was doing a lot of experiments with the lenses and mirrors and figured out that you could actually improve natural vision. So he kind of started the use of lenses for like glasses, spectacles, as they called them back then. Okay, so that was in the 1200s. And then we come along over 300 years later, and Janssen, Zacharias Janssen, and I believe he was working with his father, he found that if you put two lenses together, then it magnifies things even more. So he came up with the first compound microscope. Okay, so that was in the 1580s. After him, the next fellow that made major contributions was this guy, fellow with the interesting name. His name was Anton van Leeuwenhoek, and he was around in the 1600s. He was doing some of his work. So another, let's see, well, the compound microscope was discovered in the late 1500s, and he was doing this fellow, van Leeuwenhoek, was doing his work in about 100 years later. He is known as one of the fathers of microscopy because he built in his time over 500 microscopes, and he did a lot of work with improving lenses and resolution of the lenses. So he's known as the father of microscopy because he, through his looking at things and observing things in the microscopes and making them more powerful, he was able to bring the microscopic world to the knowledge of the people. So until then, people really didn't realize that there were these things that were living that we just couldn't see with our eyes. He, in 1673, he described the things he saw in the microscope in a drop of water as being animalcules because they had properties like animals. They could move around, and it freaked a lot of people out at the time, to be honest. He took samples from his wife and children's mouths to look at the bacteria in the plaque. He was the first to observe sperm and red blood cells and other things that we really didn't know anything about until he was able to show what he could see in the microscope. So he is the father of microscopy. Another fellow that we made note of who was important in allowing us to see great detail in the microscope is this guy. His name was William Henry Perkin, and he was a young chemist who was working under a more famous chemist in a lab situation, and he invented a purple dye. So they used the dye for many different things at the time, but in this kind of what we're talking about, the importance of it was that it could stain specimens so that you could see a lot more detail. So you could see more detail within cells or within other specimens that they were looking at in the microscope. And he was only 18 when he made that discovery, so it's pretty cool. Alright, so a quick recap. We looked at different scientists and their contribution either in developing the cell theory, so maybe they were more directly involved with figuring out that things were made of cells and that what cells looked like, or the parts within the cells, that all different organisms are made up of cells, and that cells only come from pre-existing cells. We also looked at the whole debate over whether living things came from non-living things, like Aristotle thought in the beginning from what they observed, to different people who did experiments and thought they had disproved what he thought, but people wouldn't believe them, to finally, in the end, Pasteur and his experiment with the swan neck flask and showing, no, living things really do have to come from living things. Okay, so that's kind of the first part of what we had talked about in class and why we had kind of come up with the timeline to highlight some of those important scientists along the way. Didn't cover every one of them, but did highlight a lot of the key people. So by the end of the 19th century, so that means the 1800s, by the end of the 1800s, all scientists accepted the theory of biogenesis because of the proof that Pasteur was able to provide. The microscope had become an indispensable tool, magnifying up to around 2,000 times. They could see cells and structures, and they were making more and more powerful technology. So without that development of technology, really, we wouldn't have the proof that living things come from living things. And we may still be back in the day of believing that to be true. So we move on to talk about different types of microscopes, today's microscopes, and we'll focus on the dissecting microscope, which is also known as the stereo microscope, the compound-like microscope, which you're probably familiar with or remember from Miss Ramsey's class, and then two types of electron microscopes. Now, the last day that you were actually in class, you had a substitute, and she gave you a handout to work through looking at the information about the types of microscopes and summarizing them. So I'm going to kind of talk to those features, and then we'll go over that sheet together on here. And then I'll do another little video lesson on using the microscope. Okay? So, in terms of the types of microscopes, we have the compound-like microscope, which is, again, what you would have used a little bit in junior high and what we'll use in our classes and labs that we do. Basically, you put, I'll move these out of the way. Okay, so this is the stage. You put your sample, your specimen, on a slide on the stage. The light source is right here. So light is sent up from the bottom, up through the specimen, and then it goes up through the objective lens, which magnifies it. Okay, so here's the specimen. The light goes up through, and then up through the objective lens. And then the image is carried up through the body tube, up through the ocular or eyepiece. So there's a lens there that magnifies it a second time, hence compound microscope, before it gets to your eye. Okay. It gives you a Carrie, can you turn it down too, please? It gives you a two-dimensional image, so that means flat. So there's only x and y. If you're thinking of a graph, then you have x and y coordinates, and you have a flat image of something. You don't see its depth. You can't see its surface or how it looks as a whole. It'd be like cutting through something. You see a little section of it. Okay. You can see live or dead specimens. So if you had some if you had some pond water, for instance, and you put it onto the microscope, then you might, by chance, see some living organisms that are in it, depending on your sample. But you can also look at prepared slides of dead specimens. Lots of different specimens that you can look at and observe that are relatively low cost for what we do in school. We get up to 2,000 times magnification, so you get fairly high magnification but low resolution, meaning that you can't see as much detail. So you can make it bigger, but you can't see a lot of detail within it. Okay. The second type looks like this, most of them. It's called a dissecting or stereo microscope. Basically, the light shines down onto your specimen. The specimen is down here, so maybe you're dissecting a worm, or you're looking at a butterfly, or you're watching as something alive is moving around in a little tray. Maybe it's an insect of some sort, a beetle or whatever. So you look down through here, and the light would be shining down onto the specimen. I'm not sure why that arrow is there. Anyway, I'm going to get rid of that one. Okay, so basically you see the whole object for what it looks like. You see all around the surface of it. You can't see inside of it unless maybe you would cut it open and put a piece of it there. But you can't see through it. Okay, usually with the light microscope, you can see through it if it's something that's cut thin enough, like if it was a piece of potato or a piece of carrot that was cut really super thin, and you can see through it. But for a dissecting microscope, then the idea is that you're going to look at the specimen and observe its features all over it. Okay, so that gives you a 3D image, so three-dimensional. You can see kind of, oh, I can see that this is the top of it, it's closer to me. I can see that this is the bottom of it, it's farther away, that sort of thing. So again, you can use live or dead specimens. These ones are approximately similar cost to a compound light microscope, depending on the power of it. It doesn't magnify very much. I believe most of them are up to 100x or 100 times. And it's pretty good for larger specimens. You can't fit very big specimens into the other types of microscopes. Electron microscopes are, well, first of all, different in that they use electrons instead of light, which gives them a higher resolution. It allows us to see a lot more detail. Unfortunately, it also means that specimens must be dead. And extremely expensive. Okay, so here's a couple of pictures of electron microscopes. Let's see. In this case, the specimen is in the middle. This is an electron beam being sent down through the specimen, so it transmits through the specimen, and then it's picked up by a detector and either sent through to a person's eye or sent to a computer. So if it transmits electrons through the specimen has to be super thin. Again, it gives you a 2D image, and we call it transmission electron microscope. In this one, here's the electrons. They come down, it's what sends the electrons. Okay, it sends the electrons in a beam to be reflected off the surface of the specimen. So here's the specimen at the bottom, and the electrons bounce off the surface in all directions, and they're picked up by a detector so that it gives you a three-dimensional image. Okay, it gives you an image of what it looks like all over its surface. And that one, again, has to be dead and treated in a certain way. Okay, so the first one we talked about, transmission electron microscope. The electrons go down through the specimen. Here would be the specimen here, and it's detected by a special detector at the bottom, or sent to your eye, or sent to a computer. So here's a picture of an older one. This one's huge. Would cost probably, I don't know, like at least the amount of a car. Some of them are even more expensive, like towards to $100,000. So yeah, they're expensive. We wouldn't have them at school, but they do have them at UPEI for special research projects. It was invented first in 1932. This is just for interest. I'm not going to expect you to know this particular history, okay? But at that point, it was only 400 times magnification. The interesting part is that Canadians made it much more powerful. Only 5 years later, they made it 7,000 times magnification. So it was kind of neat that Canadian researchers are able to improve on the microscope resolution power and magnification. Now you can change this actually. Let me see. A pen. Anything. Now they have them upwards of 10 million times magnification, which is pretty amazing. You can see a lot more detail now than we used to. But in order to see that detail, the specimens must be dead and they must be super thin. So they slice them, they have this tiny, it almost looks like a meat slicer, like you could see at a butcher or in the deli part of the grocery store. But it makes the specimens really, 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 really super thin. And then they're embedded in wax, they're sliced, and then they're treated with different stains so that you can see the detail. So this is an example of, this is the head of a tiny little worm, and you can see each of these, let me see if I can, that little highlighter. Okay, so each of these little guys is a bacterium. So each of those is an E. coli. You can see them eating them. So it's pretty cool. This one is showing the inner parts, so the organelles that you'd find within a cell. Okay, so this is Golgi apparatus, looks like flattened pancakes. This is a lysosome. It's filled with things that it's breaking down right now. But for a simple microscope, like what we would have, compound microscope at school, all we would be able to see is the nucleus in a cell. Okay, we can't see that much detail. We can't see the different organelles. But in this case, you can see a lot of detail, which has allowed researchers to learn a lot more, whether it's from medical research or learning about parts and functions of different organisms. You name it. It's really, really allowed us to learn a lot more about the microscopic world and cell parts and how they function, and if something's wrong, what's going wrong, things like that. The other type of electron microscope, a little more detail about it. Again, electrons are used in order to visualize the specimen. They are fired down onto the specimen will be down here at the bottom, and then they reflect in all different directions. Okay, and they're picked up by a detector and sent to a viewing screen. So this is a picture, this would be the microscope, and then sent to this computer over here. This is an example of an image you get using a scanning electron microscope. So scanning, think S for scanning and S for surface. It scans the surface. So it shows you can kind of see, okay, well, this antenna is closer to me, this one is farther away. It gives you a three-dimensional view, really detailed three-dimensional view. This is, I believe, the head of an ant. Okay, you can even see the little hairs that are on it. Pretty neat. In order, again, it must be a dead specimen, and it must be coated in gold so that you can have the electrons bounce off of it. So this is kind of a good one to show the differences in what you would see in the different types of microscopes. So this is all looking at a grain of pollen. The first one is showing in a compound light microscope. So you can see a whole bunch of different pollen grains. You can see the nucleus, but that's about it. You can't really see a whole lot of detail within them. This one is a transmission electron microscope, and it's only at 5,500 X. Okay, but you can see the detail. So these are both two-dimensional. You can't really tell what the whole surface of the thing looks like, but it's kind of like a slice through it. What does it look like? Okay, so this is showing like almost like a cut through it, and you're looking at the section of the pollen grain in more detail. So depending on where the cut or slice is at, you might see certain features of it, and you might not see certain features of it. Okay, so if you're slicing through a kind of a cell, then maybe you'll see a mitochondrion, but you might not see a mitochondrion because there might not be one in that particular section, for instance. Okay. This one is showing what you'd see with a scanning electron microscope. So that's the type that scans, it's coated in gold, and it scans the surface of it. So this is what pollen grain looks like if you could see the whole thing close up. It's pretty neat that you can see so much detail. Okay, so in your assignment, you'll have some questions asking you, okay, if you wanted to see wanted to look at this type of a sample, what kind of microscope would you use? So if it was something that was moving, that would have to be one of the light microscope that we talked about. If it was that you wanted to see the whole thing, or if you were dissecting it, or if it was that you wanted to see the three-dimensional unit of it, maybe you were looking at looking the digestive tract of something, or what have you, then you would want the dissecting or stereo microscope. But if you were looking and you wanted to watch it swim around and it was really tiny, then you would probably be using the compound light microscope. If it was something small and you wanted to see a really small feature on it, or if it was so if you wanted to see the hairs on the ant, then you would need a scanning electron microscope. So it scans the surface, but it gives you super fine detail. But if you wanted to see the fine parts within something, through a slice of it, then you would need to use transmission electron microscope. So if we wanted to look at the endoplasmic reticulum, or if we wanted to look at something else in finer detail, then we would need to use the transmission electron microscope. Okay, so come back to this one. This one was one that you were assigned to work on in your group. So if you look at that information and you were filling this out, Mac, there's one up in our room. Let me go check it there. Okay, so the first row is talking about what source is used in order to produce the image. Does it use light or does it use electrons? Obviously, if it's called compound light microscope, then it must use light. Okay? The dissecting or stereo microscope also uses light. The light shines down on the object. Okay. These ones says right in the name of them. Okay, and these ones use electrons. So there we go. It's the first part of the table complete. Now, the next part. I'm just gonna do this. Okay, can you use dead or live, and does it have to be treated? Compound and dissecting microscopes. You can use live or dead specimens. Okay. Sometimes they might use staining, but it depends on the specimen. Okay. For the electron microscopes, they must be dead. They have to have special treatment. Transmission, the electrons have to go through, right? It gives you a two-dimensional, like a slice through them. So these ones are embedded in wax and thinly sliced. Okay. The scanning electron microscope, they have to be dead too, but they're scanning the surface, and so the surface has to be coated. They have to be coated in gold, right? Now, does it give a 2D image or a 3D image? Okay, compound microscope gives us a 2D image. Can't really see three-dimensionally what the whole thing looks like. The other one that gives us 2D, you might think, oh, this one's light, it must too. But no. The dissecting one allows us to see the whole organism, or the whole, if you're looking at part of an organism, whatever, you can see the top and bottom of it. You can see all around it. So that is actually a 3D one. Transmission, the electrons go through a thin section of it. So it gives us a two-dimensional image, but much more detail than the compound light microscope does. I'm not sure what happens to the font in the program. It's doing funny things. So since you can see the top and bottom, kind of positionally where these different things are, the dissecting and scanning electron microscopes give us 3D images. Okay, 3D images, so you can see all depths of it. Okay, a scanning electron microscope gives you a much more magnification and resolution. You can see more detail. Approximate magnifications. I was looking up to see the more recent values for these. What's going on with the font? It's looking funny. Compound light microscopes up to 2000x. Okay, dissecting up to 100x. Remember, they kind of, it's better than using a handheld magnifying glass, but it's not going to give you as much detail as other microscopes. Transmission now up to 10 million times. That's pretty awesome, pretty amazing. And scanning electron microscope, so that's where we're scanning the surface, up to 500,000 times. So I don't necessarily expect you to memorize the values, but to understand, okay, so the dissecting microscope, it doesn't give us a lot of magnification, okay, but it gives us enough so that we can see more detail when you're dissecting or when you're looking at the external features of things, okay? The compound light microscope, it does magnify things, but not nearly as much detail. It doesn't give us nearly as much detail as if you had a transmission electron microscope. Scanning one, not as much magnification as a transmission electron microscope, but you're looking at the surface of it. Okay, you're looking at the 3D image. Cost-wise, well, we can afford the first two types. Not very many of them, but relatively low cost. These ones, you would only really get to see if you ended up doing some research project or if you got to go on a tour and have a look. They probably wouldn't let you touch it unless you were actually working on a project because they're so expensive. Like, yeah, super expensive. Now, the last one, I had suggested that you look at this and do a similar sketch. Okay, so if we looked at these, well, obviously, if they have electrons as their source, then they're going to be either the scanning electron microscope or the transmission electron microscope. Okay, so in this one, the electron source, both electron beams are aimed downward. In this one, it goes through the specimen, so the specimen has to be super thin. It gives us a 2D image, so it transmits through the specimen. This would be the transmission electron microscope. This one, it bounces off the surface, so the surface has to be coated in gold. Gives us a 3D picture, okay. Here's the top of it, what it looks like, kind of along the sides. It gives you a 3D view of the surface of it. Alright, the two light ones. This one, well, that's supposed to be a little frog. Not great drawing. That's supposed to be a little frog. So it would be dissecting microscope. The light shines down on the specimen and get reflected up through objective lenses towards the eye. So it only magnifies at a certain amount. Then, last but not least, this is the one we're most familiar with, where the light comes up through your slide, up through the objectives into your eye. That would be compound light microscope. Okay, so very basics of the different types of microscopes, how they work, how they kind of compare when you would use what type in general. Okay, so you could do a little sketch just like those ones on here, keep them straight. You could also make yourself up little questions like, oh, compare and contrast. How are these two similar? How are these two similar? How are they different? Great ways to keep them straight. Review them on Quizlet. Another way. Okay, so that's it for this video lesson. And I'll do another one where it just talks about use of the microscope. And we'll be moving on when we get back to class. We'll be doing an introductory lab using the microscope. So hopefully it'll help.